Welcome, everyone. My name is Mathieu Roy. I am assistant professor in the Department of Psychology here at McGill, and I'm going to be the host for this second event for our mini-series on the brain. We are located on the downtown campus of McGill's University. We are on the traditional territories of the Haudenosaunee and Anishinaabeg nations. And McGill has long recognized and honored these nations as the traditional stewards of the land and waters that we work on and meet on to exchange and learn from each other. To introduce our speakers on this evening are the student ambassadors, Adeline Lee and Anouk Arsenault. So Adeline Lee.
Okay. Thank you, Matthew. So hi, everyone. Tonight, we are very happy to welcome Professor Precup and Professor Kennedy to the Mini Science series. Today's talk is titled The Brain and AI, Frontiers of Neuroscience. Professor Precup is a co-director of the Reasoning and Learning Lab at McGill School of Computer Science. Her research covers a wide range of topics, including Markov processes, computational linguistics, and machine deep learning. Professor Kennedy and his lab looks at a variety of neuroscientific topics as well, including the molecular mechanisms behind neurodevelopment and the plasticity of mammalian nervous system. In today's talk, we are going to see where and how Professor Precup's and Professor Kennedy's research join together to provide new insights into the growing field of artificial intelligence. So without further ado, Professor Precup, I hand it over to you.
Thank you very much for that kind introduction, and very excited to be here today. Let me just share my screen one second. So it is my pleasure to tell you a little bit about machine learning, which is my field of research, and also to talk a bit about sort of the role of machine learning within the big AI puzzle. So artificial intelligence has inspired many generations of researchers and scientists who think about robots and androids and all kinds of fun things like this. But the AI really is made up of many different pieces like decision-making, reasoning, search, and machine learning has been really the key piece in this puzzle. There's different kinds of machine learning, and the one that you probably know most about or have heard most about is something called supervised learning, in which we have some input data and some desired output, and we train a model in order to predict the output on new inputs. So for example, we might have a task like trying to figure out if there are any faces in an image or how many faces there might be in an image. In this case, we might get a bunch of images with correct labels. In this case, we might get this image and we might have a label that says there are 3 faces. We would take this data and use it to train a model that can look at a new image and make predictions about the desired label. Now, really the revolution in machine learning in the last decade or so has been driven by a technique called deep learning, which at a very superficial level is inspired by biological learning in the brain. Deep learning has models that consist of many different neurons, artificial neurons. These are very simple functions like linear functions with a threshold, for example. And we train these models in order to mimic the desired outputs on input data. So for example, if we want to label images, the inputs might be a representation of the image as a collection of pixels, and the output might be the desired label that we want to put on this image. And then we would train essentially the connections between all of the very simple neurons that we have. These connections are parameterized, and we use gradient descent, a method which is really from back in calculus, in order to adjust these parameters. Now, interestingly, when we look at what these networks do, we can see some information that is akin to what might happen in a biological network as well. So for example, we're looking here at convolutional neural network. If we look at early nodes in this network, We have diagonals and nodes that detect lines at different orientations. This is somewhat similar to the early visual cortex. And some intermediate nodes might have sort of higher level concepts that are associated with them. So for example, if we want to recognize faces, you might see some neurons that light up when there's a face in the image. Now, there's also in this case a cat neuron in this network, which lights up when there's a cat in the image. Why is this? Well, because in the training data that was used for this network, there are a lot of cats because this is data from the internet. So really the concepts that are reflected in these neural networks are actually coming from the data that we used to train them. And this is also why it's very important for this data to be both of good quality and to represent the task that we really want to solve. Deep learning has brought about a revolution in many different subtasks. For example, in image recognition around 2015, training on a very large data set called ImageNet has brought convolutional neural networks to human level performance in terms of recognizing many different types of objects and multiple objects in the same image. And really, I want to emphasize that a lot of what we have learned in the last little while is how to scale some ideas that are actually 2 decades old in order to run them with more data and with faster computational devices such as graphics processing units or GPUs. Now, neural nets can be used for lots of different purposes. Here is an application for learning to generate images. And you see here a progression in the last decade or so, going from some very blurry images that kind of look like faces, all the way to faces that have a great amount of detail. Of course, this is a little bit unsettling because you know, these are fake faces, they are not real people. And so you can see how a network can actually fool you pretty easily. And so this is one of the things that, for example, ethicists of AI think about quite a bit. These are what sometimes people call deep fakes. Another application, very recent from DeepMind, is AlphaFold. This is a program that uses deep neural networks in order to predict the way in which proteins fold. So in this case, the training data is really protein sequences associated with a 3D structure that is known, and the neural network learns how to take new protein sequences and fold them. And this is actually work that's been quite well received in the community and has led to hundreds of thousands of new proteins being folded and being made available to the scientific community. Now, we talked about supervised learning, where we have input and output that are paired and paired correctly. At the sort of opposite end of that, there's unsupervised learning, where we just have data and the algorithm is trying to find interesting patterns in the data. So for example, this is data taken from an accelerometer in a mobile phone. You see that there's some periodicity. You see that some trajectories are smooth and others are not. And so we might just want to train the algorithm to, for example, understand what's the probability of new trajectories and whether these might also be cyclical or smooth. It's a harder problem because there's not necessarily a single correct answer, but there's actually been a lot of progress made in this problem too, especially in the last couple of years with very large models, deep neural networks that look at text and images and try to understand and build representations from these. So one of the models that's been very well publicized is GPT-3 from OpenAI. This is trained on a lot of text and can be used to generate text such as tweets and poems, but also to generate computer programs or mathematical proofs, or even to take a textual description like, you know, I would like a chair shaped like an avocado, and actually generate images that correspond to this. So there's actually a lot of richness in these deep models that can allow them to really do surprising things to a good degree of quality. Now, I work in a field called reinforcement learning, which is in some sense in between supervised and unsupervised learning. And it's quite close to models of animal learning from cognitive science. So in reinforcement learning, you imagine an agent that's situated in an environment, it perceives that environment, it can take actions in the environment, and as a result, it obtains a reward signal. So on one side of this image, you see a little mouse, a biological system, which is learning to press a sequence of levers. If it presses the correct sequence, it's going to receive a food pellet. And so that's the reward signal for it. And that will encourage the animal to do that sequence again. And actually animals are very quick at learning these kinds of tasks. We do the same kind of model in artificial agents, where we might have an agent that's situated in an environment that is simulated. It can take actions and it receives positive and negative numbers. And its goal is actually to maximize the total amount of reward, the sort of the sum of the numbers received over time. So the agent will learn by trial and error. It's never really told what the right thing to do is. And it has to cope with various problems, including randomness in the environment, non-stationarity, that means the environment can change over time, and also delays in the reward signal. So to make this a little bit more concrete, consider a game like Go, where you have two players. The players play on a board, and their goal is to win the game. So the perception of a reinforcement learning agent in this task would be the state of the board encoded as, let's say, 1 or -1 if there's a white or a black piece in any particular position, or a 0 if there's no piece. The actions of the agent would be legal moves. And the reward is only received at the end of the game. It would be a +1 if the agent has won the game and -1 if it lost. So, okay, we can encode this problem as reinforcement learning, but can we actually solve it? And actually the answer to this is yes. And there's been a tremendous success about 5 or 6 years ago with this program called AlphaGo and its successor called AlphaZero. These are programs that take this reinforcement learning problem, play games against themselves, and can optimize this return function to the point of becoming world champions among both computers as well as people. And the other interesting aspect is because these programs train by trial and error and are never told exactly what to do, they have the capability of inventing new ways of playing, which seem superior to those of people. Now, what's exactly inside of an agent like this? There's two kinds of representations that the agent maintains. One is called a policy network. The policy network tells what to do next. And the other one is what we call a value network. The value network, or the value function, evaluates the current position, and basically in the case of a game like Go, evaluates the probability of winning. And both of these are going to be trained from the agent's experience. Now, what are the principles for the training? All machine learning is actually driven by prediction errors of various sorts. But in reinforcement learning specifically, the algorithm makes predictions at every step about what it's going to receive in terms of rewards. And we're going to use the prediction error in order to drive the learning. The prediction error looks at the agent's prediction between one time step and the next time step and tries to ask, has the situation improved in my estimation? If so, the action that I just picked must have been good. And so we're going to improve its chances of being taken. So its probability is going to raise. So this is just to tell you there's some math behind it. I'm not really going to go very much into the details, but this is an algorithm called temporal difference learning that was proposed back in 1988 by Rich Sutton. And it's basically this idea that we can look at the estimated value at one time step and then the estimated value at the next time step, compare them, and the error between these two is what's going to drive the learning. Now, temporal difference errors has been used as a way to explain the activity of dopamine neurons in the brain by Wolfram Schultz and Peter Dayan and others. And this is a cartoon of what this looks like. So here we have activity from these dopamine neurons that are recorded individually. So each row in these graphs with the dots is one particular neuron that fires. And we're going to train using a reward signal. The reward signal is the sort of R point. And if we have a system that is, just going along, does not really know anything about the reward signal, whenever the reward signal appears, these dopamine neurons sort of get all excited and they fire, okay? And so in the top left graph, when you see the R line, you're gonna see a little activity happening, more activity happening in these neurons. And the top line shows you the cumulated activity over time. In the middle, we're looking at conditioning. And so we have a stimulus that predicts the reward. And we're looking at the system once it has had experience. And so the stimulus has been learned, and the system has learned that the stimulus actually predicts the reward. So what happens in this case is as soon as the stimulus comes on, the dopamine neuron activity rises. Why? Because these neurons anticipate that there will be a better state, that the reward is going to be received. And so they get excited, not at the time of the reward, but at the time when the reward is already predicted. Now, in the last row, we have a situation where the reward is predicted, however, it does not arrive, okay, because it's sort of not been provided at this time. And so what you see is at the time of the stimulus, there's activity, but then when the reward does not come, this activity gets depressed and dies down. It's because at this point in time, we have a negative error. We were expecting something good to happen. The situation was much worse. We have a negative error. And so these dopamine neurons essentially encode these prediction errors, these temporal difference prediction errors. And this is what you see on the right-hand side are the predictions from a temporal difference model that correlate very well with the biological data. Now, there's different ways to do reinforcement learning. One of the perhaps most popular architectures is what's called actor-critic. Here, again, we have the policy or the way of choosing actions represented and then the value function being represented. Value function is trained by this sort of error signal, and it informs the policy. It tries to tell the policy what should be the actions that will be taken. And there's been, again, evidence from neuroscience, fMRI evidence, that the dorsolateral striatum implements an actor, and the ventral striatum implements something like a critic that drives the learning. Now, what I'm really interested in my own research is going from these ideas in reinforcement learning to the broad space of AI. How can we use reinforcement learning to actually build abstract representations of the kinds that people use? You know, things like objects and causal relationships between them in the world, and actions that take place over very long periods of time, and correctly assign outcomes to them. This is something that is actually hard to do in the classical reinforcement learning framework. That framework is much more thought of as using rewards to drive immediate behavior. But actually, we can take the reinforcement learning ideas and generalize them, broaden them a little bit, and go to much more interesting things. Now, when I say interesting, I might not mean exactly what most people think of as interesting in the context of AI. You probably think, Go and text generation and smart agents that talk to you. I think about cooking. And not just because, I might be a little bit hungry right now, but because really cooking is a very complex activity that almost any person can do very well, yet AIs are really terrible at, right? And robots are terrible at. And that's because it involves different levels of reasoning and abstraction. Some of them are very high level, like choosing a recipe or getting groceries. Some of them are medium level steps, like getting a pot and putting ingredients in the pot. And some of them are small, like moving your wrist in order to stir in the pot. And then there's a lot of unexpected things that can happen, right? Like something can spill, the cat can be on the table eating your food, the doorbell rings. There's all kinds of unexpected events that the agent has to react to while still maintaining its course of higher level reasoning and understanding, things like boiling temperatures and, objects hitting each other and so on. So a lot of stuff needs to be seamlessly integrated. And we don't really know how to do this in reinforcement learning. One idea that I've been pursuing for quite a long time is what we call hierarchical reinforcement learning. So here there's an idea that an agent can reason over abstract actions and consider modeling those actions as well. So the name for an abstract action of this sort is an option, because an agent can choose it. And an option really is something like a policy or a way of behaving that starts and stops. So for example, if you have a robot, instead of thinking in terms of its individual motor torques from microsecond to microsecond, this robot can think, oh, I can use my sensor to look in front of me. If there is nothing in front of me, then I could consider a policy of just moving forward. And this policy I already know. And I'm going to move forward until I'm too close to an obstacle, in which case I stop and I reassess the situation. So hierarchical reinforcement learning has existed for about 20 years, but really it's become very popular again. It also has its own arguably neural correlates in the brain. So on the top picture here, you have a schemata of the actor-critic architecture that we described. We can extend this architecture to think about reasoning in terms of options and models of options. And in this case, more areas of the brain become implicated, like the orbitofrontal cortex and the prefrontal cortex, interestingly. And so some of the areas that are perhaps correlated in people with higher level reasoning can be thought of as reasoning in terms of these more abstract models and abstract actions. So I'm going to conclude by saying machine learning is really driving the AI revolution. There are different kinds of machine learning, like supervised and unsupervised and reinforcement learning. And reinforcement learning in particular has been a bridge between computer science, neuroscience, and cognitive science. And the algorithms and the ideas have really developed at the intersection of these fields and continue to develop there. And we use hierarchical reinforcement learning as a natural way to model and solve large problems. I can tell much more about hierarchical reinforcement learning, but it would be hard to do without a bit more technical detail. So I will not try to go in that direction. One of the things that I'm very interested in, though, as an open question, is building agents that continue to learn effectively many different skills and predictions over time. Right now, reinforcement learning algorithms thrive when we have one well-defined task, like Go. We can solve the game of Go very well, but if we asked an agent to do many different tasks with one brain, that would be very difficult, even if we are not so ambitious as to have the agent be a world champion. If you ask one agent to play with one brain, something like Go and chess and poker, that's very hard to do. And I would really like to understand how we get our AI agents beyond that barrier. So with this, I will stop and I will hand it over to Tim.
Super. Thank you very much. And wow, that was a wonderful talk. Absolutely fascinating. I will share my screen. Okay. So I'm a cellular molecular neuroscientist, and as a cellular molecular neuroscientist, the subjects that I'm going to be talking about are quite different from the previous talk. I'm going to take this towards plasticity and talking about synaptic plasticity. The image that I have here on my opening screen is a developing spinal cord, and what I use this for is to illustrate the notion that my lab is interested in mechanisms that regulate neuronal development, how the nervous system initially develops. And then if I click forward to this is actually a mature hippocampus. This drawing shows a mature neuronal network and actually activity flowing through that network. And we're particularly interested in how the mechanisms that direct the early development of the nervous system subsequently influence its function in the adult and then have an impact on neurodegenerative disease and aging in the nervous system. So that's where the talk will go. What I want to illustrate, though, is that we have, in my lab, we're actually users of deep learning and artificial intelligence. And a couple of years ago, we published this paper, which is is titled Deep Learning for High Throughput Quantification of Oligodendrocyte and Sheath at Single Cell Resolution. We used this technique as a way of analyzing and quantifying a particular type of neural cell in culture, and that's a myelinating glial cell. So oligodendrocytes make myelin in the nervous system, and oligodendrocytes are the cells that die in multiple sclerosis, and myelin is the insulating wrap around the connections, the axons that connect neurons. And when you lose that insulation, essentially there's a short circuiting in the nervous system. So we're studying these cells, and we're studying, I'll just give this as, I'm actually going to go to neurons, but I want to talk briefly about myelin. We're studying this in what's called a nanofiber assay. These are synthetic fibers that mimic axons. And you can see that these oligodendrocytes that have been placed onto these fibers are actually wrapping them. Now they have very complex geometries, and if I jump forward to this is actually live imaging of an oligodendrocyte that is wrapping a bunch of these fibers. And these are 2-minute intervals between frames, and it's imaging over 8 hours. You can see that they're very, very dynamic. They're extremely complex. And if your task was that you wanted to, say, look at a whole bunch of potential drugs and characterize how those drugs or those compounds affected the wrapping of these fibers so that you could identify things that would promote oligodendrocyte survival and promote myelination, with something as complex as this, that would be quite a challenging task. So what we did was we developed a workflow that used automated imaging and what is called a UNet architecture to train a deep learning algorithm to quantify oligodendrocyte ensheathment. And in particular, what this image shows is that as we went through, as you move through the neural network, you can actually extract images of how the neural network is looking at these structures. And here we're looking at different levels of the neural network. But when you get to the end of it, you can see that what it's doing is it's pulling out those segments, and the myelinated segments. Now, in particular, we were able to associate those individual segments with single cells. So we can then go to one of these cells, and the network is sufficiently trained that it recognizes that all of these ensheathed segments belong to this particular cell, and it gives us all the numbers associated with these segments. And we can use this now for high throughput analysis and imaging of these types of cells in culture. So that's one of the applications that we've used this for in the lab. Now, that was, Dr. Precup gave us a really tremendous introduction to neural networks. And I'm going to say a little bit about neural networks, but I want to move as quickly as possible to real neurons. So in this cartoon, we have a cartoon here of a real neuron. There's a neuronal cell. It looks like it's a pyramidal-ish cell, so like a cell from your neocortex or your hippocampus. And it has a bunch of dendrites. So here's the cell body. Here's a whole bunch of dendrites. It's got multiple dendrites. And from those multiple dendrites, they take inputs from other cells, and they then generate an action potential. The action potential travels along the single axon. And you can see this is a myelinated axon here. These are the myelinated insulating wraps. The action potential travels along the axon. It moves to synapses that synapse onto other cells. And then these cells would then be like the dendrites here, and they would pass that signal on further. Now, when you look at a neuronal network like this, so here we have a very simple artificial neuronal network, and we have a bunch of inputs. The inputs come to the input layer. There's a hidden layer of neurons here, and then an output layer here. When you see those multiple inputs coming into one cell, one way to think about that is that that's like the dendrites of this neuron here. So we have multiple inputs coming into one cell, and then that cell performs a computation which results or doesn't in generating an output. And you can think of generating a signal or not having a signal. The absence of a signal is still a signal. So that will then propagate through the network and then result in the output here. Now, if we think of that as the multiple inputs as being like dendrites and the single output as being like an axon, then these connections, the weights of those connections are like a synapse. And as was described in the previous talk, as a network is being trained against, say, some sort of ground truth data set, the way the network learns is to update those weights. It's changing the weights of these connections to then match as best as possible the ground truth data set. And success in learning means less error in that network. So what I'd like to stress is that adjusting the weight of those connections in the network is analogous to synaptic plasticity, the idea of changing the synapses in our nervous system. And that's where I'm going to take this talk. So here's a real neuron in culture. This is a cell from my lab. And as I described with that cartoon neuron, this neuronal cell has got, it's been in culture for about 4 days. It came from, it's an embryonic rat neuron. There's the cell body. It has one axon coming out here. This axon is growing out and reaching out to make connections with other cells. It's got multiple dendrites extending out here, and eventually an axon from another cell will connect with this guy. If you think of our brains, if you think of a human brain has about 90 billion neurons. And this approximation, give or take a few billion, 90 billion neurons. So that's around a million neurons. That's an easy way to remember it. In your head, you've got about a million neurons. If you like exponentials, that's 10 to the 6 times 10 to the 6. So it's a 10 with 6 zeros after it times a 10 with 6 zeros after it. And that adds up to 10 to the 12 neurons. That's an awful lot of neurons. If you think of the synaptic connections between neurons, an average neuron might have 1,000 synaptic inputs. So 1,000 synapses coming into those dendrites, the dendritic tree. Some will have less, some will have more. If you take 1,000 synapses per neuron and 10 to the 12th neurons, then you're looking at about 10 to the 15th synapses. So that's a 1 with 15 zeros after it. So that's an awful lot of synapses. That's a very large number of connections. It's a very complicated object, and we all have one. We think about the scale of this. So cells are small. This diameter of this neuron here is about 10 microns. So a micron is 1/1000th of a millimeter. So this is about 1/100th of a millimeter in diameter. If we think of synapses, synapses are even smaller. So here are cortical neurons that have been in culture now for about 2 weeks, and they are making, they've been in culture long enough that they've made a synaptic network. And we've immunolabeled that with antibodies. What you can see in red is a protein named synaptophysin, and that labels the presynaptic side of the synapse. And in green, we've got GluR1, which is a receptor for a neurotransmitter, and that labels the postsynaptic side. And my little note down here says synapses are really small. So if I zoom in on these guys and blow them up, what we've got there is the presynaptic side. So this is the synaptophysin, which labels the synaptic vesicles, which contain the neurotransmitter that are released into the synaptic cleft and signal to the dendrite on the postsynaptic side. And this is the receptor GluR1 for the neurotransmitter glutamate. And you can see that when you put them together, they're adjacent to each other like this, which is what you'd want to have happen. You want to have the presynaptic input adjacent to the postsynaptic specialization. Okay. Now, this is my cartoon of CNS, the synaptic morphology of a synapse in the central nervous system. And this is the presynaptic terminal that was red in the previous image. And this is the postsynaptic specialization. This is a postsynaptic spine. And what's cartooned into this are these neurotransmitter vesicles. These release the neurotransmitter that when an electrical depolarization, the action potential comes into the presynaptic terminal, the neurotransmitter is released. It then travels across the synaptic cleft and it binds to receptors on the postsynaptic side, this very specialized accumulation of receptors here in this dendritic spine. It's a very, very interesting structure. Now, I wrote at the bottom here, synaptic plasticity, and a key element of synapses in the central nervous system is that they're plastic, and plastic means that they can change. So you can change the strength of a synapse in many, many different ways. Imagine if you released more transmitter or released less transmitter, if you had more receptors on this side or fewer receptors on this side. And I'll give you an example of one example of molecular synaptic plasticity and its consequences that we've developed in my lab. But that's a cartoon of a synapse. I've been mentioning plasticity. I've been throwing around this word plasticity, and we can just say a little bit about where the note of plasticity comes from. And this term was first used in respect to psychology by the philosopher and psychologist William James. And he described this in the Principles of Psychology in 1890, talked about it in terms of behavior. And what he said was that plasticity is describing a structure that's weak enough to yield to an influence, but strong enough not to yield all at once. In other words, plastic in the sense that it can give, but it doesn't just completely disappear in the process. Now, if I bring this very much home and bring it to McGill, Donald Hebb was a psychologist at McGill. He was in the psychology department. He was also spent some time at the Montreal Neurological Institute, where my lab is. And in 1949, he published a book called The Organization of Behavior. And in that book, he described what's now called the Hebb synapse. And this is associative plasticity. It's that when a presynaptic terminal fires, when it succeeds in activating the postsynaptic side of the cell, he proposed that synapse would then get stronger. And this has been colloquialized as saying cells that fire together wire together. So this idea that there's an association between the presynaptic activity and the postsynaptic response. And the incredible thing when you think about that is that if you have activity flowing through your nervous system, activity will tend to reinforce more activity. It's like if you walk across a patch of green grass, you will tend to make a path. Other people will tend to follow that path. So that path will then get written into the grass. And that's how he was thinking about activity and experience changing the nervous system at the synaptic level, and then generating memory. Okay. Because Donald Hebb was at McGill, Dr. Brenda Milner was also attracted to come to McGill. And she came as a psychologist and then developed neuropsychology while she was at McGill. Dr. Milner is one of the most famous members of the McGill faculty, I would at this point. She's actually 103 years old. She's still with us. Brenda Milner is most famous for having worked with a patient named HM, who for many, many decades was only known as HM in the literature. We now know that he was Henry Molaison. He was a patient who had intractable epilepsy and was treated for his very severe seizures by bilateral removal of the hippocampus on both sides of his temporal lobes. As a consequence of that surgery, his seizures were greatly reduced. However, when he woke up, he had severe anterograde amnesia. Now, the interesting thing about that is that he's one of the most interesting patients in the history of neuropsychology. All of his long-term memories were intact. His present working memory was also intact. What was missing was his ability to move new experiences into long-term memory. Through Dr. Milner's studies of HM, she did absolutely groundbreaking studies that provided just absolute evidence for multiple memory systems in the human brain, for short-term memory, for long-term memory, episodic memory, procedural memory, that these were all different systems in the human brain. One of the key elements for episodic memory is the hippocampus, and that's illustrated here. And I'll come back to the hippocampus, but this is an ancient cortical structure where it receives input from the neocortex, comes into the dentate gyrus here. These little neurons send synapses onto CA3 neurons here. These send synapses onto CA1 neurons here, and then they run back to the cortex. So you've got this loop that runs from the cortex through the hippocampus and then back to the cortex. Absolutely essential for the transmission of new episodic memories into long-term memory. Now, in my own lab, I introduced this. I started this talk by saying that one of the things, a key thing that we're interested in, is how mechanisms that regulate neural development contribute to the day-to-day function of the adult brain. Now, a protein that I've spent a good part of my career working on is a protein named Netrin-1 and a receptor for Netrin-1 named DCC. And what this illustrates here is the early embryonic spinal cord. This is a drawing that I showed in the very opening slide of the talk. This is from actually the 1890s, drawn by Santiago Ramon y Cajal. And Cajal proposed in the 1890s that a structure here at the ventral midline of the early embryonic neural tube would secrete something that would make a gradient, and that the axons of developing neurons would follow that gradient towards the midline. And this is an image that we published in 2006. And what it shows is the protein Netrin-1 that is made by the floor plate at the ventral midline. It makes a gradient. You can see there's less and less red as you go dorsally. And these axons that grow to the midline and cross and make a commissure absolutely require Netrin to be present in order to extend and then cross and make a commissure. Now, Netrin, the receptor for Netrin that the neurons are expressing that respond to this gradient is called DCC. And if you take away Netrin-1 or you take away DCC, the nervous system is massively disrupted. The commissure doesn't form, the corpus callosum that links the two sides of the brain together doesn't form, etc. There are many, many disruptions, and the embryos actually die either at birth or just before birth. Now, having said that they die at birth or just before birth, one of the curious things is that these things, so that says that they're absolutely essential for normal neural development, for the nervous system to put itself together. If you look in the adult brain, everyone who's listening to this talk right now is making Netrin-1 and DCC in their own brain. This is actually a rat brain. The human brain is the same. This shows DCC. Here's the neocortex. Here's the hippocampus, that beautiful sort of two horseshoe structure that was in the picture with Dr. Milner. And you can see that Netrin-1, or in this case, DCC, are made by all of these neurons. So it's a very simple question. Why these things that are required for the nervous system to put itself together in the first place, why would the adult nervous system, which is already wired up, it's already got all the connections that it needs, why would neurons continue to make these proteins? And one question was, might it have something to do with synaptic plasticity? So we looked in the adult nervous system, and the first question we asked is, where are these things localized? Where are they present? This shows a dendrite, hippocampal dendrite. We've magnified it enormously here, and these are the dendritic spines along that hippocampal dendrite. So these are the postsynaptic specializations for my little cartoon here. And what you can see in black is where DCC is localized. So this is an antibody that's binding to DCC. And you can see that the heads of these dendritic spines are just decorated with DCC. There's lots of DCC all over the tip of these dendritic spines. We've actually separated out postsynaptic specialization, and DCC is enriched in the postsynaptic specialization. So it puts DCC right here in the postsynapse. If we look here at Netrin-1, so Netrin-1 is the protein that binds to DCC. It's also in dendritic spines. This is a picture from just a couple of years ago. It's a slightly different technology. It's a super resolution type of imaging. What we see here is a fluorescent dendrite, and all these dendritic spines, all these postsynaptic connections. Imagine it's invisible because it's not labeled, but there would be a presynaptic connection connecting to all of these dendritic spines. When we look at where netrin is, we can see the spine here, and if we make the surface of the spine translucent, what we can see is there's lots of netrin in green actually inside the spine. So what that says is that there's netrin in here, inside the dendritic spine, and there's also DCC on the dendritic spine, the receptor here. Now, seeing that this netrin protein is inside, this makes us immediately wonder if activity at the synapse might actually bring it out. Might that bring it out of the postsynaptic element and bring it into the synapse? And remember, this is a protein that's required in development to direct axons to make appropriate connections. Okay? So this is an experiment that was done by a postdoc in the lab named Stephen Glasgow. And what it shows is that he wanted to ask the question, is it possible that netrin is released by activity from dendritic spines? And what you can see here is he's engineered a dendrite to express a fluorescent red protein, and it's a protein that's enriched at synapses. What you can see is that red protein, which is Homer, is here in the tips of these dendritic spines. And he's also put in a version of netrin-1 that when it's inside the cell, it doesn't fluoresce, but when it comes out of the cell, now it fluoresces. So he activated one of these, he put in a solution that activated these synapses. And what you can see is that at minus 0.1 second here, we've got just the red Homer labeling the synapse. And then at 0 seconds, as soon as he applies the netrin, or as soon as he applies the activating agent to the dish, suddenly the green netrin pops out of the synapse. So activation of the synapse here is releasing netrin locally. And now we're marking that synapse as having been activated. What you can see here is that this is a time-lapse image where we're just following the same space over time. And you can see that the protein comes out and it stays there at the synapse. Now, there's a type of activity-dependent plasticity. Activity-dependent synaptic plasticity was discovered in the early 70s, named long-term potentiation. And the idea with long-term potentiation is that if, like a Hebb synapse, if you activate a synapse, does the synapse get stronger? Does activity at the synapse now make that connection stronger? And we wanted to ask, is it possible that netrin-1 release from a synapse by activation would actually enhance the activity of that synapse in a lasting way? The way Stephen did this was we had generated a loss of function mouse. This is a mouse that has a genetic manipulation in it that allows us to delete netrin-1 just from neurons. And with that mouse, what we do is we let the mice grow up normally. They have perfectly normal levels of netrin-1 in their brain, but then as the brain is maturing, netrin becomes deleted just from neurons in the forebrain. So the mice are hooked up normally. They survive in adulthood, and we're able to make slices of hippocampus, shown here, that we then either stimulate and record from in a dish. And we can measure the activity of the neurons that are activated by this input, the CA3 neurons coming into the CA1 neurons. So he wanted to do a loss of function experiment. He used these mice where we can delete netrin-1, and that was the first experiment that he did. What this shows here is that we've got synapses. He's measuring the baseline level, and then for 1 second, he stimulates at 100 hertz, at high frequency, stimulates that synapse. And what you can see is that the baseline level is here, and then normally we get because of the activity that ran through that circuit, now we have an enhancement of the strength of that synapse. However, and that's in a normal wild type mouse, not a netrin mutant mouse. In a mouse that has had netrin specifically taken out of neurons, what happens is we see an initial increase, but then that drops out. And in order to have this persistent, lasting increase in synaptic strength, this says that we must have netrin made by neurons. Netrin's required for this. Stephen then did an experiment that I genuinely never thought was going to work. And this surprises me to this day. And that's that he took a wild type slice, a slice that has normal levels of netrin-1, normal levels of DCC, and he didn't provide high frequency stimulation. He just washed the netrin into the bath where we're keeping the slice of tissue alive. And what you can see, so the bar here is 5 minutes of netrin applied to that slice. And what we see is as soon as he starts applying netrin, there's a substantial increase, a substantial lasting increase in the strength of those synapses. So this says that netrin is required for this type of activity-dependent plasticity, and it's also sufficient for this type of activity-dependent plasticity, which is remarkable. Now, I mentioned that one way that you can strengthen a synapse is that you can insert more receptors for a neurotransmitter into the postsynaptic side of the synapse. And we're actually able to assess that. That's what's shown here. This is again a dendrite that's been labeled with this protein Homer to label the postsynaptic side. You can see the dendritic spines here. And if you look at this dendrite before we stimulate it, we've got 1, 2, 3, 4, 5, maybe 6 synapses. And if we look at where we've similarly got a glutamate receptor that only fluoresces when it's on the surface of the cell. So this is the neurotransmitter receptor. Currently, it's marking 2 synapses. And we're just going to add netrin to this neuron and then see what happens to these glutamate receptors. So if we do that, there the netrin's being added. And within a few minutes, what you can see now is if you look at this glutamate receptor, this neurotransmitter receptor, we can now count 1, 2, 3, 4, 5 synapses where it's clearly the case that now there's more glutamate receptor that's been inserted into those synapses. And we've confirmed this biochemically in other ways, but one thing netrin is doing is when it's released from the synapse is it's moving more receptors into the synapse, and by putting more receptors in, now you have more ability to respond to that presynaptic signal, that presynaptic input. When you put more receptors into a synapse, the synapse actually grows in size, it changes in size. And what we show here, we did this in collaboration with Anne McKinney, who's in the McIntyre building at McGill. And her lab specializes in visualizing the structure of synapses and the structure of dendritic spines. If you look at, you can see that synapses are not just on or off. They're not digital in that sense. We've got many different sizes and shapes. We've got little thin postsynaptic spines. We've got big, thick postsynaptic spines. We would predict that the bigger ones are more mature than the thin ones. And if you focus on this thin dendritic spine, when we add this protein netrin to the dish, what you'll see, there'll be a dot that comes on. So the netrin is now added, and now we watch what happens. And within half an hour, we've got that dendritic spine swelling up, more receptors coming into it, and this would now be a more sensitive dendritic spine. And we tested that similarly using the electrode method. And what Stephen showed is here he's stimulating, and we wanted to see if synapses would wake up. If when we add netrin to those synapses, would they actually wake up? And what he did was he stimulated at a very, just at the threshold of the ability to evoke a response. And in this particular plot, the evoking a response means going down. So here, most of the times he stimulates, nothing happens. A few times it does, but almost nothing happens. But then he adds netrin, and he's stimulating the same inputs. And now suddenly, much more activity, much more activation. What this indicates is, as I've shown in the previous couple of pictures, netrin being released from the synapse by, the model would be that netrin released from the synapse, that is now moving glutamate receptors into the postsynaptic element. It's moving in these receptors. The synapse is now more sensitive to the neurotransmitter input, and we're getting more responses. So the model that what I've been describing here, as the title shows, is this is a new molecular mechanism regulating synapse function and plasticity. Now, our model is that when the synapse is depolarized, part of that depolarization, the depolarization by presynaptic activation, causes netrin to be released. When netrin is released, it signals back to the synapse through its receptor, and it activates biochemical mechanisms that move more receptors into the synapse. And that this type of activity-dependent release of netrin-1 is a key mechanism that promotes the maturation of synapses by recruiting neurotransmitter receptors into the synapse. We're working in the lab now, looking at this same mechanism in the context of a number of neurodegenerative diseases, et cetera. But this is what I'd like people to imagine is that if you remember this talk tomorrow, if you remember what we did tonight, one of the reasons for that is that your synapses are changing, and they're changing in this activity-dependent way that is actually changing the biochemical and physical structure of your nervous system. I've been showing you these high-resolution pictures of synapses and zooming in on dendritic spines, but I'd like to show this cerebellar Purkinje cell just as an example. This is one of the cells in the nervous system that has the most synapses. This is about 10,000 synapses per cell. When you think of the computation that a neuron is doing and the number of possible inputs to a cell, but then all this huge dendritic tree that is covered with all these dendritic spines, and then one single axon, just the level of subtlety that comes out of this is just extraordinary. Now, the width of that little synaptic cleft, I've been talking about sizes here a lot, and the width of the synaptic cleft is 20 to 30 nanometers. So it's a tiny, tiny little gap. I looked up today I was looking at microprocessors, and the news that I saw was that in 2021, IBM announced that they had now made 2 nanometer resolution chips, and each chip contains 50 billion transistors. Now, a transistor is nowhere near as sophisticated as a neuron, not even close, but 50 billion, we're getting into the range here where we might start thinking about the 100, if we're talking about a million neurons and 90 to 100 90 to 100 billion neurons in the brain. This sort of hardware technology is actually maybe starting to, we're getting into that level of numerical complexity, although there's all sorts of subtlety built into synapses that are not being represented here. I'd also like to show you this picture. This is an image from what's called a brainbow mouse. These mice were developed by Jeff Lichtman and Josh Shanes at Harvard. This is a mouse that allows you to fluorescently label subsets of neurons. So this is neocortex. You can see all the many layers. The many neurons are not labeled here, and what this lets you do is see the trees in spite of the forest, but you just have this phenomenal complexity. So when we think about this many synapses per neuron and this kind of complexity in a neuronal network, it's really an extraordinary thing to try and decipher and a fascinating thing to work on. I also just want to add with a little, or end with a little bit of science fiction here. So Garry Kasparov is probably familiar to many people, and he's a chess grandmaster, former world chess champion. He's very famous for the interactions with IBM and with Big Blue and Deep Blue, and the chess tournament in 1997 when Deep Blue won the tournament. When I was a kid, my father said to me that we would never see a computer beat a human at chess. So this was an extraordinary moment to actually see this happen. We would never see a computer beat the best human at chess. That turned out that was something that fell in 1997. I'd stress that that was in Dr. Precup's talk. She was describing AlphaGo, and AlphaGo is a deep learning algorithm. In this case, Garry Kasparov was playing against a programmed computer with extraordinary dedicated hardware and an incredible group of chess experts, extraordinarily talented programmers, and really, you can think of that as he has subsequently come out and said things like, the best person and a superb computer, that's the best team. And I like this quote that I saw from him recently, that he said, it's time for humans and machines to work together. That's a relatively positive way to think about the advances in artificial intelligence and machine learning, et cetera, that we're seeing. There's an awful lot of people who have darker predictions. Kasparov is actually an optimist in that sense. This made me think of something, though, which isn't what he meant. And I'm going to talk about this, though, which is that just this week on CBC, there was a news item that said brain implants allow paralyzed man to communicate, says study. And the study, in fact, came out this month. And the title of the study is a spelling interface using intercortical signals in a completely locked-in patient. Now, these are studies that came from John Donahue, who's one of the people who built BrainGate, and Bierbaumer, who's in Germany. And what they did was they were working with a patient who has amyotrophic lateral sclerosis and is completely locked in. So he's unable to communicate with his family. And they put 2 electrodes like this into his motor cortex, and they taught him to actually spell words. And he was able, through that brain-machine interface, was able to communicate with his family and talk to them, and not talk, but communicate through writing. And writing using his motor cortex to move cursors, et cetera, and activate these electrodes, which is then converted into a signal that is understandable. And through that, he was actually able to communicate with his family in a way that he hadn't been for ages. Now, these are 3.2 millimeter microelectrode arrays. They're Utah arrays, and they're tremendous devices. But one of the limitations of these devices is that when you put them in a brain, they're recognized as a foreign object, and that foreign object generates scar tissue that then starts to seal off the electrodes. So they work extremely well, but they don't work forever, and they tend to move, which means they need recalibration essentially on a daily basis. Now, one thing that we're doing as part of the neuroengineering program at McGill is we're working to develop a more biological brain-machine interface. And the idea here is we know now an enormous amount about synaptic biochemistry, the molecules, the polymer, the proteins that hold synapses together and can actually enhance synaptic strength, et cetera. We're working with proteins, with engineered polymers, with synthetic polymers to engineer synaptogenic surfaces that have enhanced biocompatibility in the central nervous system. What this shows is a 7-micron diameter bead that is actually coated with one of our synaptogenic synthetic polymers. And what you can see are in the red here, these are synapses that are differentiating onto the surface of that bead. And what we'd like to envision is developing surfaces that are not recognized by the nervous system as foreign objects, that are actually integrated into the nervous system and that neurons will make a connection to. As we see, what I'd like to try and communicate is that as we see that there's more and more interaction between humans and machines, both in let's say a social level and also on a medical biological level, what we want to do is enhance the biocompatibility of those interactions and facilitate it using these types of approaches. So I would thank my lab. This is the folks in the lab. I like this photo because this is very pre-COVID. And this young woman here, Jeanne Madrange, is actually continuing the work that I described that Stephen was doing with synapses. And I would acknowledge this guy, Stephen Glasgow, who did all of a huge amount of the synaptic plasticity work that I described, And I'm very happy to say that Stephen started his own lab at University of Guelph in the Department of Biology just this year. And with that, I'll say thank you. And I will stop the share. There we go. And we're ready for questions now.
Yeah. Okay. So that were two fantastic talk. So yeah, thank you very much, Professor Precup, and thank you, Professor Kennedy. Okay, so the first question is for Professor Precup. We just heard that the interactions between human and machine are becoming more and more common these days. In your opinion, like the human brain seems to have have developed this preference for social behavior over the course of evolutionary history. So we were just wondering, like, in your opinion, do you think that's something AI can also develop, but just within a very short period of time?
That's a really great question. And indeed, there is quite a bit of research in the AI community in the area called multi-agent systems. That means, either AIs that work together or AIs that might be in a loop with humans or other kinds of agents, maybe pre-programmed agents. And they can train in order to collaborate well, for example. From the point of view of sort of modeling the problem, it's in some sense all the same. You can imagine an agent that's a reinforcement learning agent. It perceives the environment. The environment contains other agents, and its reward might be dependent on those other agents as well. So for example, it might receive gifts from other agents, or maybe they're working as a team, and so the team receives a reward, and that reward gets split evenly between the members of the team. And indeed, we see these agents developing collaborative behavior. We can look at whether they're developing communication protocols. So for example, we can allow them to say things to other agents, and they can learn how to sort of come to an encoding of what they want to say. So I definitely believe that this is possible, and we have some of the tools that are needed in order to train agents to collaborate with others. I think it's the main problem at the moment in that area is that, just like in any kind of situation where you have an ensemble, you end up with an equilibrium, but not necessarily with the best solution possible, right? So this comes from game theory, right? And from the work of John Nash, and we see it play in human society. It also plays out in AI society, where, the ensemble of agents might end up in a solution where nobody wants to do something different, yet maybe that solution is not what we intended it to be. And so I think there is a lot of interesting research going on at the moment to try and understand how do we design these kinds of equilibria where the agents can, work well together in the sense that we expect them to. And for example, one of my undergraduate students did a wonderful project a couple of summers ago, looking at agents rewarding each other, so exchanging gifts in some sense, in order to, for example, induce behavior like cleaning up, which nobody would want to do otherwise. And these kinds of ideas can actually work quite well.
Yeah, that's perfect. Thank you.
Yeah, thank you so much for that wonderful question, Professor Precup. Professor Kennedy, we also have a question for you. So you compared how input passes through a neuron system in a human brain with how it passes through an artificial neural network. So considering that, do you think that computation in the brain is digital?
So the short answer, the really short answer would be no. The longer answer is it depends what you mean by digital. And if you think of digital as a concept, so like alive and dead, that's digital. It's two things. So in the nervous system, what's usually accredited with being digital is the action potential. So the signal that goes down an axon is kind of either there or it's not. What's beautiful though is when you get to a synapse, do you remember the green dendrite with all the different bumpy synapses on it? So there's so many different gradations of synaptic strength. So you have all these synapses, and then within one synapse, it's a whole universe in that little synapse, and all these different gradations of strength. And that's not digital. That's many, many, many different degrees. I'd also, I sort of hinted at it, but one of my favorite quotes is from Gregory Bateson, who said, he said that the absence of a signal can also be a signal. And the example he gave was not sending in your taxes. And so it's when we think about digital signaling, when we think about signaling through a network, the presence of a signal and the absence of a signal are both extremely important. So I'll stop there.
May I ask a follow-up question to that? Because it really struck me in your wonderful talk that the plasticity itself is mediated from all kinds of sources, right? Like these chemicals, right, that are there. And in artificial neural networks, we just have the gradients. The gradients flow wherever there was input, right? There's nothing that's protected in some sense, and there's nothing that becomes more plastic. And so I'm wondering if you see an opportunity to take some of the ideas, some of the biology, and somehow maybe model it more accurately in neural networks, in artificial neural networks.
One of the things, I'd be very curious to talk more with you about the reinforcement learning that you're doing, because that type of reinforcement pathway can be extremely important for synaptic plasticity. And we could talk about synaptic architectures that would support that kind of reinforcement through things like other neurotransmitters coming in, like dopamine, and reinforcing the strength of the synapse. And then when you go back in history and you think of like and this is not definitely not my field by a long shot, but when I think of something like the perceptron and then the Boltzmann machine, and I think the Boltzmann machine was one layer. And I'm old enough that I remember when that was published, and I remember being very excited about it, and I thought it was just the coolest thing. And then they took the one layer and they made two layers, and adding depth was a huge step forward. And when you look at the nervous system, there's depth all over the place. And then feedback was another big step. And the first circuit I ever studied in the nervous system was a little feedback loop. And it's you can look at the history and you can say, here's how the architecture of the biology perhaps is inspiring what's going on in the neural networks. But today the neural networks are getting so sophisticated that I actually wonder the opposite. And to what extent are the learning rules coming out of these incredibly sophisticated neural networks that can do extraordinary things? So.
Well, so thank you very much, Dr. Precup and Dr. Kennedy. Thank you for the great presentations and the very interesting discussion. So I would now like to thank the speakers officially with a gift. So you are going to receive postcards from the Red Pat Museum. So it's a stereoscopic postcard. So when you open it up and look through it, you're going to see this amazing dinosaur in 3 dimensions. So the Red Pat Museum is Canada's oldest museum. It is located at McGill. It was opened in 1882, and it used to be open to the public, but unfortunately it is currently closed. But they are still working, and we are also sending you some of the museum's publication, which are accessible at the gift shop. So you have access to the museum's exhibit through the book. So for instance, here we have all of the minerals that are in the museum's mineral gallery. And the last part of your gift is a face mask. So it looks like we'll probably need to continue to wear these for some time, and so you can add this one to your collection. Finally, the Red Pat is also running a fundraising campaign. So the outreach team is currently building discovery boxes where instead of schools coming to the museum, they can send these boxes in schools and in remote communities and underserved communities. And we would really like to have these boxes full of amazing things like minerals, fossils, skeletons, bones, etc. So this campaign is running as part of McGill 24 crowdfunding, It ends on May 12th, and all donations are welcome. So thank you again for coming in tonight. We'll see you next week, Wednesday, April 6th, for a presentation on memory, models, and philosophy of the brain with Drs. Ian Gold and Brendan Johns. So thank you again for coming in today, and thank you, Dr. Precup and Dr. Kennedy.